In this presentation, we will talk about the liver, as it is one of the most vital organs we cannot live without, and the different types of hepatitis that affect the liver's normal functions. The liver breaks down carbohydrates, proteins, and lipids, detoxifies blood and hormones, synthesizes plasma proteins and blood clotting factors. It stores glycogen, fats, iron, and copper, secretes hormones, removes bacteria and worn out blood cells. The liver is the largest organ in the body, weighing approximately 3 pounds, which explains its multiple functions that it performs. But what happens when the liver stops working because it is infected by a virus like hepatitis? Hepatitis is inflammation of the liver and may be caused by a variety of factors, such as infections or toxins. Hepatitis can occur when one of these variables overwhelms the liver's functional capacity, resulting in injury, damage, or destruction of cells. Acute hepatitis may totally resolve and never return, but chronic hepatitis does not resolve and usually continues to progress. There are 6 recognized viruses that can cause hepatitis. Transmission of the virus and symptoms that one may experience vary widely according to the type of hepatitis, but the beginning stages are all the same. Infection will occur when the virus invades and attacks healthy cells of the liver. In sequence, the immune system will respond with inflammation and attempt to fight off an infection or heal an injury. But continued inflammation left without treatment can permanently damage the liver, as it will start to scar healthy liver tissue. This is known as fibrosis. As excess scar tissue grows, it cannot do the work that healthy liver tissue can. This will block blood from flowing through the liver, causing the normal function to deteriorate over time. This is known as cirrhosis. Hepatitis viruses are a diverse group of viruses that share remarkable tropism or affinity for the liver. These viruses are profoundly different from one another in their physical structure, epidemiology, and mode of transmission. Nonetheless, these viruses find the liver to be a major site for them to replicate. When hepatitis A enters the body, it goes into a 2-week incubation period before any initial symptoms of the disease appear. Hepatitis A is a limited disease that does not result in chronic infection. Antibodies produced by the immune system in response to a hepatitis A infection last for life to protect against reinfection. Hepatitis B is extremely infectious and spreads through bodily fluids, contaminated needles, or may even be transmitted from mother to unborn child. Approximately 90% of infected infants become chronic cases, comparing that with 2 to 6% of adults with chronic cases of hepatitis B. Hepatitis B can lead to cirrhosis or liver cancer. Hepatitis C is spread mainly through contact with infected blood, including IV drug use, needle sticks, and unprotected sexual contact. Hepatitis C can be a short-term illness, but a long-term chronic infection can result in serious health problems that lead to death. Hepatitis D is very infectious and strongly associated with intravenous drug abuse. Hepatitis D occurs only in conjunction with hepatitis B, as it is needed for activation and reproduction. While there are vaccines to prevent hepatitis A and hepatitis B, there are no vaccines available for hepatitis C, E, and G. Although hepatitis D can be prevented with hepatitis B vaccine, those who develop chronic hepatitis B are later infected with hepatitis D. Hepatitis E produces acute symptoms. It is a self-limited disease that does not result in chronic infection. The virus can be severe for pregnant women during the 3rd trimester, as they may develop a severe illness or acute liver failure. Hepatitis G is a recently discovered viral form that is transmitted via the blood, typically believed to occur alone or in the presence of hepatitis B or C, which in this case is referred to as a co-infection. The severity of the disease depends on countless factors, cause of inflammation, as well as any pre-existing physical conditions. Blood tests will measure elevated bilirubin and aminotransferase levels, and the presence of antigen and antibody factors. Liver biopsy is required to confirm chronic hepatitis and determine types and degree of damage from liver disease. Ultrasounds and liver CT scans are used to diagnose cirrhosis.